I want you to imagine a scenario for a second. You're a military commander, right? And you are fighting a war against this really deadly, heavily entrenched enemy.
Okay, yeah, like a standard siege situation.
Exactly, a classic siege. So your strategy is to just starve them out. You know, cut off their supply lines, block all the roads, and just wait for the inevitable collapse.
Right, you just wait for them to run out of resources.
Yeah, but, and here's the twist, instead of dying out, that starvation tactic accidentally acts as like a brutal evolutionary pressure cooker.
Oh, wow.
Yeah. The intense pressure actually forces the threat to adapt, to mutate, and it becomes stronger, faster, and infinitely more prone to spreading far behind your own lines.
I mean, that is just the ultimate strategic backfire, isn't it? You deploy this tactic thinking it's going to guarantee a win, but you're actually inadvertently fueling the enemy's most aggressive evolution.
Right. And that biological backfire is exactly what we are unpacking in today's deep dive. We are exploring the incredibly complex and honestly totally counterintuitive world of clear cell renal cell carcinoma.
Which, just for shorthand, you'll hear us refer to as ccRCC today.
Yes, ccRCC. It is the most common and the most lethal form of kidney cancer. And our mission for this deep dive is to decode a massive clinical mystery that was outlined in a fascinating 2026 paper from the journal Cancer Cell.
It really is a dense, incredible paper.
It is. We're basically trying to understand why standard frontline treatments explicitly designed to starve these tumors of blood, why do they eventually fail, and, how do they sometimes even provoke the cancer into this deadly metastatic rage?
Well, the sheer volume and the quality of the data we're looking at to answer that is just remarkable.
Oh, absolutely. The source stack is huge.
Yeah, they didn't just look at one biological angle. The researchers used genetically engineered mouse models, specifically these KVPR models, designed to perfectly mimic human treatment-resistant kidney tumors.
Right, so we aren't just looking at random cells in a petri dish.
Exactly. And on top of that, they utilized single-cell RNA sequencing to track the gene expression of literally hundreds of thousands of individual cells.
Which is mind-blowing.
It is. And then, maybe the most impressive part, they employed high-definition human spatial imaging.
Oh, I love this part. It's literally mapping the battlefield, right?
Yeah.
Hey.
It shows us exactly where every single immune cell and blood vessel is located in the three-dimensional space inside the patient.
It's like having, satellite imagery of a microscopic war zone. So we're going to break down every mechanism, every piece of jargon, so you walk away with a crystal clear understanding of this tumor microenvironment.
Let's get into it.
Yeah, let's start at the baseline. Before we can even talk about why the cure fails, we have to understand the fundamental architecture of the disease itself. What is actually going wrong inside a ccRCC kidney tumor before any drugs enter the picture?
Well, the entire disease state is basically built on a genetic illusion.
An illusion? How so?
So the fundamental biology is driven by the loss of a specific gene called VHL, which stands for von Hippel-Lindau.
Okay, VHL. What does it normally do?
In a healthy cell, the VHL protein acts as a highly sensitive oxygen sensor. Its main job is to regulate a pair of proteins called HIF-1-alpha and HIF-2-alpha.
HIF-1-alpha and HIF-2-alpha. Got it.
Right. So when oxygen levels in your body are normal, VHL essentially tags these HIF proteins so the cell can destroy them. It just keeps everything in a nice balanced state.
But in this kidney cancer, that doesn't happen.
No. In ccRCC, there is a biallelic inactivation of the VHL gene.
Meaning both copies of the gene are broken, the genetic instructions are just gone.
Precisely. The cell has lost both copies. So that VHL oxygen sensor simply doesn't exist anymore. The sensor is completely blind.
Oh, wow. So if it's blind, it can't tag those HIF proteins for destruction.
Exactly. And because of that, the HIF proteins rapidly accumulate and sound a massive systemic alarm inside the nucleus. They act as transcription factors, literally rewriting the cell's behavior.
Okay, so the cell is panicking.
Total panic. The tumor cells enter a clinical state known as pseudohypoxia. They act like they are suffocating and starving for oxygen, even when they are sitting in a perfectly oxygen-rich environment.
That is, it sounds exactly like a building's fire alarm system malfunctioning. The alarm is just screaming at the top of its lungs, triggering the emergency sprinklers to flood the building, even though there's no fire anywhere.
That's a perfect analogy.
Okay, let's unpack this a bit more. If the tumor thinks it's suffocating, what is its equivalent of turning on the sprinklers? Like, how does it react to that fake alarm?
It initiates a process called hyperangiogenesis.
Angiogenesis being the creation of new blood vessels, right?
The fake hypoxia signal forces the tumor to frantically build a massive tangled network of new blood vessels. It's desperately trying to bring in more oxygen to fix a suffocation that isn't actually happening.
But hold on. If a tumor is panicking and hastily throwing together emergency plumbing overnight, are those blood vessels actually functional? I mean, tumors are notorious for having chaotic, leaky, terrible vascular networks. Is this rapid-fire construction just a defective mess?
You know, that's exactly the assumption a lot of researchers had. But the spatial imaging data from this study completely proved otherwise.
Wait, really? They actually work?
Yeah. To test the integrity of this plumbing, the scientists used a specific hypoxia-binding dye called pimonidazole, or hypoxy probe.
Hypoxy probe. Okay. What does the dye do?
It's engineered to covalently bind to cells only if the local oxygen level drops below a really critical severe threshold. So they injected this dye, along with CD31 markers that highlight blood vessels, into their KVPR mouse models.
And what did they find?
They found that at baseline, before any treatments, these tumors have virtually zero true hypoxia.
Wow. So the frantic building project is a total success. They really aren't suffocating at all.
Not at all.
Yeah.
Their microvascular networks are actually highly efficient. The vessels are evenly branched, and crucially, they're fully supported by parasites.
Parasites, what are those?
They are specialized cells that wrap around the outside of blood vessels, kind of like structural scaffolding.
Ah, okay. So they keep the pipes from leaking.
Exactly. They provide stability. So the tumor's plumbing is actually excellent. It can efficiently support its own massive runaway growth right up until the physical mass becomes so huge that it just outpaces even this hyper-efficient blood supply.
Okay, let's do a quick recap so we don't lose the thread here. We have a ccRCC tumor with a broken genetic sensor. That broken sensor tricks it into thinking it's suffocating. That's a pseudohypoxia.
Right.
And that illusion drives the tumor to build a massive, functional, well-supported network of blood vessels. It is thriving on a false alarm.
Exactly. It's heavily armed and well-fed.
So if you're an oncologist looking at this well-oxygenated, rapidly growing mass, how do you even begin to attack it?
Well, you attack the very thing it just built. The standard of care, frontline treatment for this, is a powerful combination therapy known as TKIIO.
Okay, let's break this down. What's the TKI part?
TKI stands for tyrosine kinase inhibitor. In this context, we use VEGFR-TKIs, like a drug called lenvatinib.
Lenvatinib, and what does it target?
It targets the blood vessels. These drugs block the receptors that govern blood vessel growth, basically acting as a chemical tourniquet to cut off the tumor's blood supply.
Okay, so there's our starvation tactic. You realize the tumor relies on this plumbing, so you bring in a drug specifically designed to destroy it. What about the IO part? What's the second half of the combo?
IO stands for immuno-oncology, specifically immune checkpoint inhibitors, like anti-PD-1 drugs.
Ah, anti-PD-1. I've heard of that.
Yeah, they're designed to take the molecular brakes off the patient's own adaptive immune system. They empower the body's T cells to recognize the cancer as a foreign invader and actively hunt it down.
I mean, that sounds like a highly rational two-pronged attack. You chemically starve the tumor with the TKI, and while it's weak, you unleash the immune system to sweep through and clear it out.
It makes perfect sense on paper.
Right. But obviously this paper exists because that logical approach has a flaw. The long-term durability of the specific combo isn't as robust as we'd hope, right?
Sadly, no. Compared to just pure IO regimens, the TKI combo tends to burn out faster in the long run.
So why does adding a starvation drug eventually cause the immune therapy to fail?
To understand that, we have to bust a major myth in oncology regarding how these TKI drugs actually affect the plumbing. For years, there was this theory called vessel normalization.
Vessel normalization, like making them normal.
Yeah, the idea was that if you give a TKI at the right dose, it doesn't totally destroy the vessels. It just prunes away the chaotic, leaky ones, leaving behind a normalized, highly efficient network that actually improves blood flow.
Oh, I see the logic there. If you want your anti-PD-1 immune drugs to physically reach the center of the tumor, you need a functional highway system to deliver them. So they assumed the TKI was just fixing the roads.
Exactly. But the data from this paper unequivocally states that vessel normalization does not happen in ccRCC.
Oh, wow. So what happens instead?
When they treated the tumors with lenvatinib, it didn't gently prune anything. It caused total catastrophic vascular collapse.
Total collapse.
Total. The structural parasites detached, the vessels degraded, and the entire plumbing system was just decimated. And this immediately plunged the tumor into massive, true hypoxia, leading to widespread necrosis, which is actual tissue death.
Okay, time for another quick recap because this is wild. Lenvatinib doesn't fix the vessels, it destroys them. It replaces the tumor's fake pseudohypoxia illusion with actual severe oxygen starvation.
Right. It creates a wasteland.
But wait, this brings up a huge question. If the TKI is successfully choking the tumor and causing massive necrosis, why do patients eventually relapse? Like, who is surviving in that wasteland?
That is the central paradox. A tumor isn't just a static lump of cloned cancer cells. It's a dynamic, responsive ecosystem. To find out what survives, the researchers used that single-cell RNA sequencing to look at the cells navigating the necrosis.
And who was left on the battlefield?
The sequencing data showed that the tumor microenvironment was completely dominated by myeloid cells.
Myeloid cells, that's a broad family of immune cells, right?
It is, but in this specific context, the dominant players are macrophages.
Okay, macrophages. Normally, those are the heavy lifters that engulf debris, clean up dead tissue, and help coordinate the immune response. But knowing tumors, I'm guessing these aren't normal macrophages.
You guessed right. They're heavily influenced by the environment. We call them tumor-associated macrophages, or TAMs. And here is where the paper makes a structural discovery.
Lay it on me.
The TAM population isn't a monolith. There are two entirely distinct, functionally opposed types of macrophages fighting for dominance inside this tumor.
Okay, let's unpack these two opposing forces. Who is the first major player?
The first group is categorized as MM0 macrophages, also called solanify macrophages.
NM0, what do they do?
Genetically, they express high levels of genes related to antigen presentation. Functionally, these are your diplomats.
Diplomats, I like that analogy.
Yeah, their biological job is to scavenge pieces of the mutated tumor, process them, and physically present them to the T cells, basically flagging the target for destruction.
That sounds exactly like what you want. If you're giving an immune-boosting drug like anti-PD-1, you want the diplomats pointing the T cells right at the cancer.
You'd think so. But the single-cell data showed something completely counterintuitive. These MN0 diplomats are actually found in highly elevated numbers in tumors that ultimately resist the anti-PD-1 treatment.
Wait, what? They're trying to organize the attack, but their presence correlates with failure.
Yep. It's confusing at first glance.
We definitely need to figure out why they're failing. But before that, what's the second type of macrophage?
The second population is the MM1 macrophage. These are marked by specific genes like SPP1 and GPNMB.
So SPP1 positive macrophages, are they diplomats too?
No, not at all. Unlike the MM0s, these cells have zero interest in talking to the adaptive immune system. They are highly glycolytic, wound-healing macrophages.
So if they aren't talking to T cells, what are they doing? How do they relate to that blood vessel collapse?
To answer that, the researchers designed a brilliant in vitro experiment. They took human macrophages and put them inside a sealed hypoxic chamber to simulate the true, severe, low-oxygen environment caused by the TKI drug.
Oh, clever.
And they proved definitively that these SPP1 positive MM1 macrophages only differentiate and activate in response to true low oxygen. They completely ignore the tumor's baseline, fake pseudohypoxia.
Wait, really? So the tumor is screaming about fake hypoxia all day long, and these MM1 macrophages just totally ignore it?
Totally ignore it. But the second the TKI drug comes in, destroys the blood vessels, and causes actual suffocation, these MM1s swarm the area.
Okay, let's summarize this for everyone listening, because we now have essentially a cellular tracking device. We've got MM0s acting as the diplomats for the T cells, and MM1s acting as emergency first responders who only show up when the oxygen is actually gone.
Exactly, which means SPP1 positive TAMs are the perfect spatial biomarker for true tissue necrosis. If you see them swarming a zone, you know for a fact the TKI drug successfully choked the blood supply right there.
That is fascinating, but here's the thing about biology: correlation isn't causation. Just because the MM1 first responders are at the scene of the crime doesn't mean they're influencing the outcome.
Right, exactly.
So how do the researchers prove who actually matters?
They ran an ablation experiment. They asked, what happens if we just wipe all the macrophages off the board entirely?
Wait, how do you wipe out all the macrophages without killing the mouse?
They used a targeted drug called a CSF1R inhibitor, specifically BLZ-945. CSF1R is a vital receptor that gives a continuous survival signal to macrophages. Block that receptor and the macrophages just die off, leaving the rest of the tumor intact.
Okay, so the board is cleared. No MM0 diplomats, no MM1 first responders. What happens when you give them the TKI and IO combo treatments now?
The results were radically different for each drug. For the TKI, lenvatinib, The therapeutic response was completely unchanged.
Unchanged, so it still starved the tumor just fine.
Yeah, it still caused vascular collapse and necrosis, even without the macrophages.
I mean, it makes sense. The TKI is just a chemical hammer hitting the blood vessels. It doesn't need immune cells to break pipes. But what about the IO drug, the anti-PD-1?
The immune therapy response was completely abolished. The survival benefit completely vanished.
Completely abolished. Wait, if anti-PD-1 is meant to take the brakes off the T cells, why does removing macrophages cause the T cells to fail?
Because it proves the T cells are entirely dependent on those MM0 diplomats. The drug might take the brakes off the T cell assassins, but without the MM0 macrophages actively presenting the targets, the assassins are completely blind.
Wow. So you don't need macrophages to starve a tumor, but you absolutely need them if you want your immunotherapies to work.
Exactly. And they found something else unexpected when they wiped out the macrophages.
What's that?
Remember the parasites? The structural cells holding the baseline blood vessels together.
Yeah, the scaffolding.
It turns out macrophages are required to maintain healthy parasites. When they depleted the macrophages for a prolonged period, the parasites just detached on their own. The vessels degraded, and the tumor started starving, even without the TKI drug.
That is wild. So the macrophages are actively secreting survival signals to keep the plumbing intact.
Yep, everything is interconnected.
Let me do a quick recap here. This is a delicate balance. We wipe out macrophages, and we realize they actually support the blood vessels, but they also fundamentally required for immune therapies to function.
Right.
Which leads us to the ultimate clinical paradox of this entire deep dive. Is oxygen starvation actually a good thing for the patient?
Well, it depends entirely on the timing. And this was the big aha moment of the paper. If we look at patients who are actively on treatment, hypoxia is a good thing. It correlates with a good response because it means the TKI drug is doing its job, collapsing vessels and killing tissue.
Right. On treatment, starvation equals success. But what about before treatment?
They analyzed human clinical trial cohorts and real-world data, and they found that if a patient's tumor naturally has a high hypoxia signature pre-treatment, it predicts terrible outcomes.
Why is baseline starvation a bad sign?
Because if a tumor is experiencing true hypoxia before you even give it drugs, it means the cancer is so insanely aggressive that it has outgrown its own hyper-efficient blood supply. It's created this chaotic dead core where immune cells can't penetrate.
Oh, I see. It's not a sign of weakness. It's a sign that the cancer is a hardened survivor.
Exactly. It's a massive warning sign.
And this brings us to the most alarming finding in the whole deep dive. We know short-term starvation kills a lot of the tumor. But what happens if you intentionally starve the cancer in mice for a really long time?
They tested this using specialized syngeneic metastatic models, LVRCC67 and LVRCC88.
Okay, models designed to track how cancer spreads.
Right. They applied prolonged, chronic TKI-induced hypoxia. They locked the tumor in severe starvation for weeks.
And what happened? Did the cancer eventually just die off?
No, it actually exacerbated metastasis. The primary tumor in the kidney was shrinking under the drug's pressure, but the cancer was spreading far more aggressively to the lungs and liver compared to the control group.
The drug was shrinking the main mass but causing it to explode everywhere else. How is that possible? What is the escape mechanism here?
It's extreme selective pressure. You kill the weak cells, but the survivors don't just sit there. The harsh environment activates a survival pathway called TGF-beta signaling.
TGF-beta, what does that do?
It initiates a profound cellular transformation called pre-EMT, or epithelial to mesenchymal transition.
Let's break that down. Normal kidney cells are epithelial cells, right? Like rigid bricks in a wall.
Yes, tightly bound. But when they undergo this transition to a mesenchymal state, they have a physical identity crisis. They shed their rigid structure, dissolve the proteins binding them together, and become highly mobile and flexible.
So they literally mutate their physical shape.
Yes. They mutate to survive the stress, detach from the tumor, slip into the bloodstream, and migrate to new oxygen-rich environments like the lungs or liver.
That is terrifying. Short-term starvation clears the bulk of the tumor, but long-term starvation breeds a generation of aggressive escape artist clones.
Exactly. And it perfectly explains why TKI-IO combos can eventually burn out in the clinic.
Okay, as we wrap up, let me synthesize this incredible journey for everyone listening. We started with a tumor-faking oxygen starvation to build great plumbing.
Right.
We hit it with a TKI drug that actually starved it, destroying the pipes. We watched MM1 first responder macrophages rush in to clean the mess, while MM0 diplomats tried to direct the immune therapy.
Which completely failed when we removed them.
Yes. And ultimately, we realized that long-term starvation forces the tumor to evolve, use TGF-beta signaling, mutate its shape, and metastasize.
The clinical relevance of this is just immense. It suggests oncologists might need to seriously rethink chronic, continuous anti-angiogenic therapy in aggressive cases, just to avoid forcing that metastatic flare.
It fundamentally shifts how we view the battlefield. Squeeze it too hard for too long, and it slips through fingers. Which leaves us with a final, lingering question for you to ponder.
Oh, I know where you're going with this.
If targeted starvation creates these escape artists via TGF-beta signaling, could the future of cancer treatment be a triple threat?
A simultaneous attack.
Exactly. Imagine a regimen where you block the blood vessels to starve it, boost the immune system to fight it, and simultaneously deploy a localized TGF-beta inhibitor to lock down the tumor's escape routes before it even realizes it's trapped.
Weld the doors shut before you cut the power.
Exactly. Something to think about until next time.